All right, today we are cracking the code on some of the most brilliant medicines ever invented, antiviral drugs. I mean, these things are like microscopic secret agents we send in to fight an enemy that has already breached our walls. So, let's get into it.
I mean, really think about that. That is the fundamental puzzle. See, unlike bacteria, which kind of just set up camp outside our cells, viruses are the ultimate parasites. They can't do anything on their own. They have to get inside our cells and turn them into little virus-making factories. So the real question is, how do you stop them without wrecking your own body in the process?
So here's our game plan. First, we'll cover the basic challenge of fighting viruses. Then we're going to open up the case files on 4 major viral enemies to see exactly how these drugs outsmart them. And finally, we'll zoom out and look at the bigger picture, this never-ending arms race.
Okay, so here's the absolute crux of the problem. Viruses use our own stuff to replicate. They don't bring their own equipment for making proteins or anything like that. They hijack ours. This makes them an incredibly tricky target. You can't just carpet bomb the area, right? The whole strategy is to find a process, any process, that is totally unique to the virus and attack that. And that's exactly what an antiviral drug does. It's a medication that usually doesn't kill the virus head-on. Instead, it just masterfully interrupts one specific step in its life cycle. You can think of it like throwing a tiny, perfectly aimed wrench right into the gears of that virus factory, shutting it down while leaving all of our own cellular machinery running just fine.
Let's open our first case file, the good old flu. The influenza virus's whole operation is basically like a bank heist. It's got a meticulously planned strategy to get in, copy its assets, and then get out to hit the next target.
Okay, so check out the plan. The heist kicks off with entry, using a protein called hemagglutinin like a key to unlock the cell door. Then a step called uncoating, where the M2 protein opens an internal gate to release the viral blueprints. After all the replication and assembly of new viruses, you get to the final, crucial step, the escape. Another protein, neuraminidase, acts like a pair of bolt cutters, snipping the new viruses free so they can escape and start the whole heist over again.
So, how do we stop this heist? Well, our drugs are the security system. M2 inhibitors, for instance, literally jam that M2 gate shut. The viral genes are trapped. They can't get out. Then you have endonuclease inhibitors that sabotage the copying machine itself. And my favorite, neuraminidase inhibitors, like Tamiflu, they disable those bolt cutters, so the brand new viruses are stuck to the outside of the cell, unable to escape. It's absolutely brilliant.
Okay, next case file. Let's level up. HIV is a much more complex enemy. It's a special type of virus called a retrovirus, which means it has a few extra, really sinister tricks up its sleeve. And fighting it requires a much more aggressive, multi-pronged attack.
Now, the really wild parts here are steps 2 and 3. Once HIV gets inside, it uses a unique enzyme called reverse transcriptase to rewrite its genetic code from RNA into DNA. Then, and this is the scary part, another enzyme called integrase literally cuts open our cell's DNA and stitches the viral DNA right into our own genome. The virus becomes a permanent part of that cell's blueprint.
So how in the world do you stop that critical step? Well, this shows 2 incredibly clever ways to do it. The first class, NRTIs, are basically imposters. They look like the building blocks of DNA, but they're faulty. The viral enzyme grabs one and adds it to the chain, and bam! Construction stops cold. The other class, NNRTIs, are more like a wrench in the gears. They physically bind to the enzyme itself, twisting it out of shape so it just can't work anymore.
And that's just one piece of the puzzle. Modern HIV therapy, what we call the cocktail, hits the virus from every single angle. You have entry inhibitors to bar the door. You have reverse transcriptase inhibitors to jam the photocopier. You have integrase inhibitors to stop it from editing our DNA. And finally, protease inhibitors to stop the final assembly line. It's a full-on assault, leaving the virus with nowhere to turn.
On to our third case file, Hepatitis B and C. These viruses make a beeline for the liver, and they're notorious for setting up shop for the long haul, creating chronic infections. They're like these persistent squatters inside our liver cells, causing slow, steady damage over many years.
The tactics here are razor sharp. For hepatitis B, the drugs are very similar to what we saw with HIV. They target its special polymerase enzyme and terminate the DNA chain. But for hepatitis C, modern drugs, called direct-acting antivirals, are a total game changer. They are incredibly specific, targeting key proteins that are absolutely essential for the virus to replicate its RNA, but that have no equivalent in our own cells.
And it's also worth mentioning a totally different strategy.
interferons. These are a bit of a blunt instrument. Instead of targeting the virus itself, they're basically a synthetic version of our body's own alarm system. They scream to all the nearby cells to put up their defenses, creating a really hostile environment that just generally messes with any virus trying to replicate.
Now for our final case file. And honestly, I saved the best for last. The strategy against the herpes virus family, it's just one of the most elegant and ingenious bits of drug design you'll ever see. It is a perfect Trojan horse attack.
Okay, so here's the genius part. Drugs like acyclovir are given as an inactive molecule, what we call a prodrug. It just floats around your body, completely harmless. It can't do a thing to any of your healthy cells. The only way this drug becomes a weapon is if it happens to wander into a cell that is already infected with a herpes virus.
So let's just walk through this because it's amazing. The harmless drug enters a cell. If there's no virus, nothing happens, it just leaves. But if that cell is infected, a special viral enzyme called thymidine kinase sees the drug and mistakenly arms it by sticking a phosphate group onto it. Then our own cell's enzymes finish the job, adding 2 more phosphates. And this fully activated 3-phosphate weapon is then grabbed by the virus and put into its own replicating DNA, which immediately grinds the whole process to a halt. The virus is literally tricked into building the bomb that destroys it.
So, as you can see from all these different case files, the story of antiviral drugs is a story of incredible human ingenuity. But it's not a story with an ending. It is a constant, ongoing battle, an arms race.
And this quote just says it all, doesn't it? Viruses, especially the RNA ones, they evolve and mutate at a mind-boggling speed. A drug that's a silver bullet today might become less effective tomorrow as the virus just evolves around it. It's a chess match that forces us to constantly innovate just to stay one step ahead.
And that really just leaves us with this final question to chew on. We've seen drugs that can block doors, jam up machinery, and even trick a virus into self-destructing. So what comes next? Are we going to target parts of our own cells that viruses depend on? Or maybe develop even broader spectrum drugs that can fight whole families of viruses? The race is on, and you can bet human ingenuity is rising to the challenge. Thanks for tuning in.